In this video, we will discuss about recombinant glycoprotein platform. So, what is this? So, many biopharma industries, when they try to produce some kind of glycoproteins, instead of developing a new machinery on their own, the idea that most of the biopharma or all of the biopharma company use is to use already existing machinery that is present in the nature. So what they did initially, how it started with a bacterial cell. It started with a bacterial cell. So they try to produce the protein of their interest from the bacterial cell by using the machinery of the cell of the bacteria. So however, the problem that exists in this bacterial cell is that it does not have an enzyme for post-translation modification. No enzyme for post-translation modification. Post-translation modification. So, however, after some time, scientists have identified some enzyme in different organism that can do post-translation modification or N-glycan modification. So they have injected those enzyme into they have modified this cell with those enzyme and injected those enzyme in this bacterial cell. However, even then, whatever the post-translation modification occurs is not similar to human, and because of this there is a huge possibility that glycoproteins that are produced by this using this machinery might instigate a immunogenic response from a human body. So however, it is used to produce non-glycosylated proteins and very limited form of glycosylated proteins. Next platform that scientists have used is of the yeast. Yeast has its own machinery for post-translation modification. However, the post-translation modification is not similar to the human beings, and it produced mostly hypermanosylated glycoproteins, and which is different from human beings, as human beings does not produce hypermanosylated glycoprotein. So if someone inject glycoprotein produced from this yeast directly to the human beings, it will produce a immunogenic response and some kind of allergy also. So there is modification needs in this yeast cell machinery to develop the glycoproteins that are compatible with humans. And the reason why people choose yeast as it can produce, it has a very high efficiency, and this cell line is established very well. And by efficiency I mean its yields is also higher than the mammalian cells, which is the human cells. So commercially this is favorable. However, the final product is difficult to make it compatible with the human beings. So another machinery that another cell lines that people have tried is the plant cells. Plant cells produce plant cells has the N-glycosylation. Plant cells has the plant cells also face the same problem similar to yeast, is that its post-translational modification is not similar to human beings. it produce not the hypermanosylated glycans more. However, it produce high mannose or posymannose. High mannose or posymannose glycan. Posymannose glycan more than posymannose glycan. Whereas these kind of glycans are not usually found in the human cell lines, in the mammalian cell lines. So because of this, also plant cell also is not been able to use directly for glycoproteins formation. However, all these platforms use all these glycoprotein produced by all these platforms which I am presently discussing has been approved. Some of the glycoproteins produced by this platform has been approved, but that is approved by on this modified platform, not the general platform. But all this, what I mean to say is that whatever the glycoproteins that are approved using this platform has been approved on the modified form of this platform, not in the general form of this platform. Another cell lines that people use is insect cell. Insect cells. Insect cells has the post-translational modification similar to human beings. However, the problem with this is that it has some kind of epitopes that are not usually present in human beings that might be available in the final glycan structure, such as N-acetyl N-glyconumeric acid. N-glyconumeric acid and glyconumeric acid. And glyconumeric acid. So this is not usually present in the human beings. And another one is the galactose, alpha galactose. Alpha galactose. These are two examples of the epitopes that are present in the insect cells. However, it is not present in the human cell beings or mammalian cells. So because of this, the glycoprotein produced by this insect cell lines might create an immunogenic response or some kind of allergy. And one more thing that we need to understand that I forgot to tell you is that this plant cell are not able to produce silated glycans, whereas there are silated glycans, glycoproteins are there in the humans or mammalian cell lines. And same problem is with the insect. Insect is also not able to produce silated glycan. No silation occurs. No silation occurs in insects and plants. So far we have seen that no platform has been able to produce the post-translation modification that is similar to that of mammalian cells. So finally, scientists will stick to the mammalian cell lines. mammalian cell lines. So right now around 70% of the glycoproteins that are approved by FDA are produced in mammalian cell lines. So it has post-translation modification similar to human beings. However, why mammalian cell lines is not the first choice of the biopharma industries is because it is easily contaminated. Contaminated. It is easily contaminable. Contaminated. We can say that it is easily contaminated by foreign substance. And also when it comes to commercially produce these mammalian cell lines for the desired glycoproteins, the yield is not so good. Yield is not so good. Yield is not good. So that's why biopharma industry first try to choose the other options. If they can produce their desired glycoproteins in cell lines that has a higher yield, that can reproduce faster, then they will prefer those cell lines instead of going to the mammalian cell lines. So I hope you have some kind of idea about recombinant glycoprotein platform and how the biopharma industry, how and why biopharma industry use this kind of platform instead of developing their own platform. Thank you.